Hello and welcome to Nikolay's genetics lessons, and today's problem is from the F2 generation of a cross between mouse genotypes, that is homozygous dominant with homozygous recessive, one male progeny of the genotype A blank space, so it can be dominant homozygous or heterozygous, was chosen and made with homozygous recessive female. All the progeny and the resulting litter were of the dominant phenotype, but we don't know of what genotype. How large a litter is required for you to be able to assert with 95% confidence that the father genotype is homozygous dominant. How large litter is required for 99% confidence. Probably there are many different ways how we can solve this problem, but in this video I'm going to show you a method of solution of this problem that is going to be based on the Bayesian theory. And this is a mathematician of the 18th century that contributed a lot to the theory of the probability statistics, and many modern applications are based on this theory. And I'm not going to show you a formula today because I have found that it is much easier to show you just step-by-step method of table, and it is much easier for you would be to understand and apply this table method in order to solve genetics problems like this one. First of all, before I will show you a solution, I want to tell you that this type of problems only students that major in genetics probably can get, or those students that study math and statistics, or those who study bioinformatics. All the rest students, it is highly unlikely that you may get a problem like this one. But if you want to master genetics, you probably also would be interested in be able to solve such problems. So first of all, let's draw a pedigree according to this problem. We have in parental generation one female, one male. So what we know about this generation from the F2 generation of a cross between mouse genotype, that is homozygous dominant and homozygous recessive, so we don't know which mice is female, which is male, and which genotype belong to specific sex. So we don't care actually, and we can say that female can be, for example, homozygous dominant, male here can be homozygous recessive. So if we would cross them in the next generation, which is F1 generation, we may get different progeny. It's not necessary for us to show all the possible combinations and variants of the sex of the litter. Just let's take two representatives of this litter. Of course, this representative have to be of the different sex, because we have to cross them, and this is how we are going. So this is going to be F1 generation, this is parental generation, and this is how we are going to get F2 generation. Once again, we take only two representatives of this F2 generation. Those, of course, litter would be much bigger. So, what we can tell about F1 generation? It is very easy to predict that 100% of the F1 generation are going to be heterozygous. So, going to be capital A and small a. So, once again, one parent only contribute dominant allele, another parent only can contribute recessive allele to the following generation. So with 100% confidence, we can say that all F1 generation are going to be heterozygous and going to be carriers. In parental generation, this genotype would produce affected phenotype, but in F1 generation, 100% would be the same phenotype as a female parent in a parental generation, so would be normal. But what we can say about F2 generation, we build simple Punnett square. So genotype of one parent on top, genotype of another parent here on the side, and according to the Punnett square, once again, these both parents came from the F1 generation. So these two parents, one here on the side, another here on the top. We know that 100% of the animals in this litter would be heterozygous. So both sexes. Let's say that this is going to be female side and this is going to be male side. So what we can expect that 1/4 of the progeny would be homozygous dominant, 1/2 we expect to be heterozygous, and 1/4 we expect to be homozygous recessive. Let's return to our problem. One male progeny of the genotype that is dominant, but it can be, as you see, different genotype. It can be with homozygous dominant, or it can be heterozygous. Phenotype is going to be the same, just like parental phenotype, so would be normal. But we don't know whether it is going to be homozygous dominant or heterozygous. So that's why we have here genotype capital A and blank space. Because these parents here came from F1 generation, so here we are going to have a representative of the F2 generation. And we are told, according to our problem, that one male progeny of this genotype was chosen once again from F2 generation. So this is possible genotypes, and mated with a female, which is homozygous recessive. So let's underline female is homozygous recessive. So this female here in F2 generation, we know her genotype, but we don't know genotype of this male. So once again, that means that this female have to be affected, but this male here is not affected, and genotype can be whether homozygous dominant or heterozygous. And now we have to find probability that this male randomly chosen from F2 generation would be homozygous dominant with 95% confidence and 99% confidence. And we have to find this using a certain number of the progeny depending on their phenotype. So over 9 minutes I am already explaining just what this problem is about, but believe me, this is going to be easiest explanation because the guy who sent me this problem couldn't solve it even though I gave him a formula. So I'm going to use table method today, and this is going to be easiest variant how to solve this problem. Imagine now, for example, that this male is affected. So what we can say about genotype of this parent? If this male is affected, that means that recessive allele he have to get from mother side, another recessive allele, so genotype have to be small a, and another recessive allele this animal have to get from the father side. So we would say with 100% confidence that this animal is heterozygous. So as you see, nothing changed here, but just information about the progeny may change probability of this animal of being homozygous dominant or heterozygous, because our prior knowledge would tell us that if we would randomly choose from F2 generation one male animal of the dominant phenotype, probability of this male being homozygous dominant would be so being capital A, probability would be 1/3, and his probability of being heterozygous or capital A, small a, being 2/3. Once again, this is very important, not 2 out of 4 or 50% or 1/2, but 2/3, because we know that this male phenotypically normal, so his probability of being a carrier or being heterozygous is 2 out of 3. And probability of being homozygous dominant is 1 out of 3 and not 1 out of 4, because we know for sure that this male is not homozygous recessive. So I'm just explaining how additional information may change our understanding of the probabilities that have been prior to this knowledge. Now think about different variant. What if this couple of animals would have, say, 4, 5, or 6 progeny of the different sex, but all the progeny is going to be phenotypically normal. Now you understand that probability of this male parent of being homozygous dominant instead of heterozygous much higher than before, because if this parent is going to be heterozygous and we cross with another parent that is homozygous recessive, we expect that 50% of the progeny going to be homozygous recessive. And we expect it to be according to Punnett square. Once again, let me draw a Punnett square and let's suggest that this parent is heterozygous, which is probability is 2/3. So once again, female parent genotype is known, which is small a, small a. So this is female side, and here is the male side, and let's suggest that male is heterozygous. So in this case, probability for this couple to have affected and unaffected progeny would be as follows: capital A, small a, small a, small a, capital A, small a, and small a, small a. So as you see, we expect that 50% of the progeny would be affected and 50% of the progeny would be unaffected if male parent would be heterozygous. But if we would see a picture like this, that all the progeny here 1, 2, 3, 4, 5, 6 would be unaffected, this increased probability for this male to be homozygous dominant. But we don't know so far how much the probability would be increased that this male would be homozygous dominant. And base formula theory would help us to solve this problem. So first of all, we have two hypotheses here. The first one, that this parent, male parent, is homozygous dominant. That's why all the progeny is phenotypically normal. For example, if this couple would have 6 animals in a litter, then of course if this parent would be homozygous dominant, another homozygous recessive, 100% of the progeny would be phenotypically normal and would be heterozygous. But another probability also exists that this parent is heterozygous and just by chance only they have all the progeny that is phenotypically normal. Those we expect that 50% being affected according to this Punnett square. 50% affected and another 50% of the dominant phenotype unaffected. As you remember, we choose this male from F2 generation from those animals that show dominant phenotype, and here's our two probabilities of the genotype of this parent. So basically we have two hypotheses, whether this male parent can be of this genotype or this genotype. Now let's use this information. So our prior knowledge give us probability that male is whether heterozygous or whether male is homozygous dominant. So only these two variants, and this is going to be our prior knowledge. So this is prior, and this prior knowledge tells us that probability of the male of being heterozygous, as you remember, is 2/3, and probability of the male to be homozygous dominant is 1/3. So 2/3 plus 1/3 would give us 100%. So only two variants are possible. Next line would be conditional information. So conditional. And conditional information would be that this couple would have 6 progeny of the unspecified sex. Sex is not important for our calculations. Why choose 6 instead of say 20 or 15? I just choose this number because I know that it would be very close to 95%, and I already did calculations for 5 and 7, and I have found that 6 would give us numbers that we are looking for. I don't know, maybe another express method exists to be more precise about the number, but I just choose these numbers randomly based on my internal feelings that we should start with 6 animals in the last generation. So what we have here? We have probability that first animal would be affected if father is heterozygous. So this is probability A, and this is probability B. And according to this Punnett square, if we take into account that male parent heterozygous, then we expect that 50% would be affected, 50 would be unaffected. So probability that this first animal in a litter would be affected or unaffected, chances are equal. In our case, we are looking for probability of being unaffected, and the probability is 1/2. And the same probability is here, 1/2. So we put 1/2 here, 1/2 here, 1/2 here, 1/2 here, and 1/2 here. So even if one parent is heterozygous, another parent is homozygous recessive, they may have normal, phenotypically normal progeny, and as you see, probability is rather high, 50%, but when we take into account that they have 6 progeny that is unaffected, 6 animals in a litter, now we can calculate how probable this event. And we just have to multiply all these independent probabilities, and this would give us conditional probability of having all 6 animals in a litter being phenotypically normal as 1/2 multiplied by 1/2 by 1/2, 1/2, and 1/2 here. So 1, 2, 3, 4, 5, and 6. So probability would be 1/4, 1/8, 1/16, 1/32, and 1/64. Now you see it is not very probable that parent being heterozygous, another parent being homozygous recessive, would have 6 animals in a litter that is going to be all phenotypically normal, and probability would be 1 out of 64. Now let's consider if male parent would be homozygous dominant. And as you remember, if male parent would be homozygous dominant, another parent would be homozygous recessive, 100% of their progeny would be phenotypically normal and would be heterozygous. So 100% would be heterozygous, and this is what we expect. So we expect that first animal being phenotypically normal as 100%, second animal being phenotypically normal 100%, third, fourth, fifth, and sixth. So if we multiply 1 by itself 6 times, we're still going to get 1 or 100% probability of such an event if, once again, male parent would be homozygous dominant. Then we expect that all the progeny are going to be phenotypically normal. And next line in our table is going to be joint probabilities. So joint probabilities of the prior probability and new probability that we have found. And in this case, if we consider that parent would be heterozygous, our joint probability would be 2/3 multiplied by 1 over 64. And this is going to be 2 over 192, and we can simplify it to 1 over 96. And on the right side, joint probability would be 1/3 multiplied by 1, and we are going to get still the same 1/3. Any number multiplied by 1 would be the same number. And now last line that is going to be final, so final or posterior, and basically here is going to be simple formula, and here is a formula. So we have to divide joint probability of event A by joint probability of the event A plus joint probability of the event B. As you remember, event A is probability that father is heterozygous, and joint probability A is here. So we have to just put numbers here. And on the right side, we are going to have the same formula, slightly modified. So here we would have joint probability of the event B divided by the joint probability of the event B plus joint probability of the event A. So what we are going to have here on the left side, joint probability of event A is 1 over 96. So 1 over 96, we have to divide by 1 over 96 plus 1/3. So what we are going to have here on the left side, so let me draw a line here, and we are going to have 1 over 96 divided by 1 over 96 plus 1/3. So how we add 1 over 96 plus 1 over 3? Our denominators have to be the same. So we have to multiply 3 by 32, and numerator 32. So we are going to have 1 over 96 divided by 1 over 96 plus 32 divided by 96. So 1 over 96, and here we are going to have 33 over 96, which is going to equal to 1 over 96 multiplied by 96 over 33. We just change division by multiplication, and we have to put this fraction upside down. So this is what we are going to get, and now we can cross this number in numerator and this number in denominator, and the answer going to be 1 over 33. If we divide 1 by 33, we are going to get 0.033. Need an answer in percentage form. We can say that we can move this decimal point 2 places to the right, or we have to multiply this number by 100, and our answer would be 3.3%. Probability that father is heterozygous. As you remember, this side was a suggestion that father is heterozygous, and in the light of this conditional information, we have found that new probability of the father of being heterozygous 3.3%. We can do the same calculations here. I just want to save a time, and I can tell you that the number that we are going to get on this side would equal to 100% minus 3.3%. So on this side we are going to get 96.4% confidence that genotype of the father would be homozygous dominant. And as you see, prior probability was only 1/3 or 33%. And here, prior probability was 66%. So once again, let me put these numbers directly in our pedigree. So our prior probability, as you see, numbers are here. Now, if we know that this couple have 6 progeny of the, doesn't matter what the sex is, that is unaffected, we can say that probability that this male is homozygous dominant would be 96.4%, and probability that this male is heterozygous would be 3.3%. But you may reasonably ask that according to our problem, we have to find probability for this male to be homozygous dominant with confidence of the 95%. But we have found 96.4% that we got if this couple would have 6 babies in a litter of the dominant phenotype. And actually we cannot get exactly 95% because the progeny number of the animals in a progeny have to be a discrete number, so it can be 1, 2, 3, 4, 5 and not 5.5, for example, or 4.9. So this is closest number that we can get, and if we would take 5 animals, we will get a number that is about 93 point something percent, so this number would be closer to 95%. That's why I choose 6. And because this video is probably one of my longest videos, I'm not going to show you the same computation. In order to find 99%, I just can tell you that this is 8 animals that have to be of the dominant phenotype. So we can say with over 99% confidence that this animal, this male, is homozygous dominant, that is randomly chosen from F2 generation here. And once again, now you can repeat step by step my calculations, just taking into the account that 8 animals would be phenotypically normal. Actually, this calculations doesn't take so long. I can do it under 1 minute, but explanation took me so long. But actually, when you know what to do step by step, such calculations shouldn't take you longer than 1 minute. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.